Hello, my name is Ariel Gispan, and I'm a computational biologist at Immunobiotherapeutic. I'm presenting the poster titled Challenges and Inconsistencies in Type 2 CRISPR-Associated Nuclease Subtype Classification. CRISPR-associated nucleases were first found and classified as a component of the bacterial immune defense system, designed to combat foreign DNA. The discovery of SpCas9 and its repurpose as a genome editing tool led the path for the discovery of additional distinct nucleases with diverse properties that are employed in a variety of applications. In recent years, more and more diverse CRISPR systems were found and led the need to group them into distinct groups. Indeed, this field has been developing rapidly, and it's common to describe the grouping hierarchy in terms of classes, types, and subtypes. In type 2 nucleases, our group of interest, the subdivision into subtypes was based on combination of different methods, but there is no straightforward rule that will allow us to unambiguously determine the subtype of a given nuclease. In this study, we compared different classification approaches on a large-scale database of 9,500 type 2 CRISPR systems, namely loss architecture, based on the other CRISPR genes near the nuclease, Cas9 phylogeny, which is based on the Cas9 nuclease sequence, Cas1 phylogeny, based on the sequence of Cas1, which is present in all CRISPR systems. The distribution of nucleases by subtype was studied and the agreement between the methods was measured. We can see that while there is a variability in the percentage of each subtype, overall we can see that the most common two subtypes are 2C and then 2A, and we can find 2B in only around 1% of the cases. Interestingly, while we expected to find Cas1 in all cases, it was missing in around 8% of the CRISPR system that we were looking at. The classifications from each pair of methods were compared, and the pairwise consistency ranges from 70 to 80%. Overall, only 71% of the nucleases were classified to the same subtype in all three methods. Out of these consistently classified nucleases, which represent around 70% of our data, 55% was classified to 2C, 44% to 2A, and only 1% to 2B. In most cases of samples with ambiguous subtypes, it was between 2A and 2C. In other cases, new loci architecture patterns were found, missing genes reverse the direction, and in some cases, even genes that were not yet annotated as CRISPR-associated proteins. Since the HNH domain is unique to type 2 nucleases and it's highly conserved, we try to classify nucleases based only on their sequence. We use k-means for clustering HNH domain sequences, and we found that not only this can be done, but we also found subgroups within the same subtype with distinct profiles. We measure the activity levels of nucleases in mammalian cells in order to see if it's related to their subtype. We found no correlation between activity levels and subtype. We couldn't predict activity based on subtypes or even on the subgroups employed from our HNH clustering analysis. To conclude, these findings show that the paradigm of affiliating nucleases into distinct homogeneous groups cannot explain significant fraction of the huge diversity of type 2 nucleases, and most probably cannot be used in order to predict its function. We propose that newly discovered nucleases should be carefully characterized before any attempt to classify them into existing classification. Thank you for listening.